process information, cells need the right proteins for the right job. Study number 4, we're going to answer the question, how does RNA polymerase know which genes to transcribe? By know, we mean slang of how does it find and bind to the appropriate DNA in a gene, the promoter. So the investigators wanted to know what proteins actually bind to a promoter. To do this, they had to come up with a new method. So the first thing they did was they synthesized a radioactive promoter. And once again, you can see here the same promoter sequence we've seen from T7, the virus. It's a good, strong promoter. They took that promoter sequence and loaded in a gel here under the label P for promoter, and it's shown as a black box sitting in the well. So that's the DNA, radioactive DNA sitting in the well of the gel. And to its left, the molecular weight marker shown in blue. They turned on the current, the electrical current in this gel, and started the gel electrophoresis so that the small molecules migrated fast and appear at the bottom of the gel, and the bigger molecules did not migrate as far. And what you can see from this example is that the promoter, again shown here, migrated very fast because there were no proteins bound to it, so it's a small molecule. This time, they added RNA polymerase to the promoter sequence. They took this sample and put it in the lane with P+, shown here as pink. They performed the electrophoresis, turned on the current, and you can see that the band with the promoter bound with RNA polymerase migrated much slower, meaning its collective molecular weight was very large compared to the P lane where the promoter migrated all by itself, a smaller molecular weight. That's the method they're going to use, so let's look at the real experiment. They take the radioactive promoter, mix it with 4 different proteins, put it on a gel, and turn on the current. In this case, they had RNA polymerase, RNA pol, and then they mixed it with 3 different transcription factors they knew bound to this particular promoter. In the first column, we do our negative control, where none of the 4 proteins, either the 3 transcription factors or RNA polymerase, were incubated with the promoter, turned on the electrophoresis, and the promoter migrated all the way to the bottom. It looks black because it's the radioactivity exposing the X-ray film. And the next column, our positive control, where all 4 proteins, the 3 transcription factors, and the RNA polymerase were mixed with the radioactive promoter, and you can see that it did not migrate very far into the gel because that's the size of the promoter plus the 4 bound proteins. In our first experimental condition, we left out the transcription factor number 1, and you can see that the promoter migrated as if nothing was added to it. In the next column, we left out transcription factor number 2, and again, the promoter migrated in a very small size, indicating no proteins bound to it. In the third experiment, we left out the third transcription factor. Once again, the promoter is small, no proteins are bound. In the final experiment, they added all 3 transcription factors but left out RNA polymerase. And maybe it's a bit of a surprise, but again, the promoter was small, indicating no proteins had bound at all. So what we learned in this experiment is that in order for transcription factors and RNA polymerase to bind, it was a all or none response. Now this is just for one particular promoter. Not all promoters will work this way, but in this case they found that all 4 proteins had to be there in order to bind, for any of them to bind, and so they bind in an all or none fashion. So we learned in this study, case study number 4, RNA polymerase binds to where transcription factors bind. They bind all or none fashion. In case study number 3, the previous one, we found out that RNA polymerase binds to specific sequences. DNA sequences matter, not just the number of hydrogen bonds.